Thank you. That was fun. Thanks, everybody. That was great. I will talk about extinct flowers. I won't talk about photosynthetic animals today, but feel free to ask me later. I'm supposed to pull up my slides. 1 second. I'm ready to go. All right. So this is a bacterial photograph. This is a petri dish blown up, covered in bacteria that have been genetically engineered to produce pigment in response to light. So it's sort of like a photographic film, but made with bacteria. They're engineered by students from UT Austin who were competing in a competition called the International Genetically Engineered Machine Competition. It happens in Boston every fall. This was quite a while ago in 2006. The students are a lot of biologists and a lot of computer scientists come together to program biology. That's the field of synthetic biology, is bringing those two fields together and thinking about programming not ones and zeros that you would in computer code, but programming DNA code. This is the DNA code that makes those bacteria turn into film. And you can kind of schematize it using those little blops and blips to say this is how it tells the cell to do all the things that it needs to do. The sensing of the light and the production of the black pigment in response. And so the students who participate in iGEM, they contribute all of their source code to this registry of parts. It's a big library now that's in a big freezer in Cambridge, where all of this DNA code is being stored so students can reuse it and build their own projects. There's lots of really exciting things. Check out igem.org. You might ask, why would we spend all this time trying to genetically engineer microbes to do weird things like make film? The film is sort of one interesting application, maybe not super useful, but when you think about what biology can do and why you might want to engineer biology, one of my colleagues, one of the founders of Gingko, he says biology is nanotechnology that works. When you think about what's going on inside of cells at this tiny, tiny, tiny level, it's incredible. So on the left, you have the protein that plants use to capture sunlight energy and convert that into chemical energy that they can use. It's each one of those little like squiggles, that's like atomic scale, so like the level of atoms making these super, super, super precise structures. Human nanotechnology cannot even come anywhere close to this kind of precision, and this happens on its own. It just evolved. It grows in dirt anytime. And so the power of biology at that incredibly tiny scale to build really complex structures that can do incredible things, capture solar energy, and then translate that into something that happens at the global scale, the ecosystems that are all around us, and all of us, we're biology that's kind of working at all of those scales at once. If we are able to tap into that to make our technology look more like biology, something that can grow and be recycled, something that can work within the bounds of nature, I think we have an incredible opportunity to think about what is possible. So I'll give you a few examples of ways that people are currently thinking about synthetic biology and using biology as a technology. Nanotechnology, a big trope in nanotechnology is sort of the fantastic voyage, this shrinking down of nanorobots that might go in our body and find disease and cure it. Turns out biology already has these really little nanorobots. Our immune system cells that are able to kind of crawl around and capture, in this case, bacteria, and search and destroy them. People nowadays, synthetic biologists, engineers, are taking those cells, those immune cells from our body. They're able to take them out, genetically engineer them, and put them back in patients so that they can actually seek out and destroy cancer cells. That's called CAR-T, and it's something that's starting to be FDA approved and coming out there and helping patients with cancer. There's also, this is a root of a plant, so now from inside of our body to down in the soil. On the root of a plant, these are legume plants that have these little pink nodules that pop out. Those nodules are full of bacteria that provide nitrogen to the plant. So that's why you don't have to give nitrogen to things like to beans and legumes. You don't have to give them fertilizer because they're making it themselves with this really complex interaction that's happening in these tiny structures in the roots. In contrast, when we want to make nitrogen fertilizer for our plants, we have to do it in something like this. This is a chemical plant that's producing ammonium fertilizer. So when synthetic biologists look at this problem, they try to look at the parts that are inside of those cells in the bacteria that are in those roots, look something like this, and translate that into a program that can be programmed into bacteria that could go and live on other kinds of plants. So not just the beans and the legumes, but maybe something like corn. So there's a new company that spun out of Gingko that I work at, a joint venture between Gingko and Bayer called Join Bio that's working on this problem, trying to engineer the bacteria that live on the roots of plants to actually provide nitrogen to them, to decrease the amount of fertilizer that we have to add and significantly improve the sustainability of agriculture. So that's just to give you a kind of taste of like what biology can do and how people are looking to translate that incredible thing that biology does into technologies today. And then when you think about, well, what is biology and biotechnology? Biotechnology has been around for a long time. It's really about the way that we can transform things with biology, with living things. And one of the best examples of that, I think, is beer. Beer is the transformation of sugar and grain into carbon dioxide, alcohol, and various kinds of flavors that we make beer via yeast. So the yeast will consume the sugar and produce all of those different things. And so when a synthetic biologist looks at this, they think, okay, well, what can I do with the DNA of the yeast to make something else come out at the other end? And what might those other things be? So maybe you want to boost or change some of the things that are already there. So maybe you want to make more flavor or a different kind of flavor. This is a project that I've worked on, so I'll tell you a little bit about it. So take oranges. You want to make a good citrus beer. You take the genes that are responsible for making orange flavor in oranges. You sort of find that sequence. I'll send it in an email to a DNA synthesis company, in this case Twist Bioscience, a company that we work with. And a few weeks later, they'll send me a tube with freeze-dried DNA in it that has that sequence in it. And then the lab that I work in and the company, Gingko, we've built this lab that uses a lot of automation to take that DNA and transform it into the yeast. So now that yeast becomes a little factory for producing orange flavor via the DNA that we were able to engineer in it. This is a little snapshot of what the lab looks like. It's actually now like much more crowded. So this is a much older video, but I kind of changed to show you how the robots work. We use the robots because they're able to kind of expand what one person can do. So I spent a lot of my time in the lab when I was a graduate student, like moving tiny bits of liquid around. Now robots like this can do that for us, which is great. So one person can do a lot more, can make a lot of different kinds of experiments all at the same time. So in the end, you can take that yeast that now has that DNA from oranges and brew beer. So this is something I did with two of my colleagues who are really accomplished home brewers. We've found we were able to make some beer with this orange-flavored yeast. It was delicious. Not all of the brewing, most of the brewing we do is not home brewing, but it actually looks a little bit more like this. These are tiny little like micro breweries. They're about sort of soda can size, and we have these sort of large machines that can do 12 or 24 of them at once, so we can test out a lot of different conditions for brewing the yeast and trying out different things. And then from here, in our lab where we have this kind of small scale, we can send it to a lab that can grow it up in a really big brewery. And from that big brewery, you can make beer, but most of our products are actually extracted from that beer. So it's actually something that's purified, the yeast is removed, and you have a product at the end. That product might be a flavor or a fragrance. We work with flavor and fragrance companies like Robertet. to produce a number of different kinds of flavors and ingredients that can end up in all sorts of products. This technology, this kind of brewing of products, is something that has been going on for a long time. It's used in medicine and pharmaceuticals. Insulin is made this way, so you can take the gene from humans that makes insulin, put that into bacteria. The bacteria now grow insulin, and that's where we get insulin from today. It's used to make the enzymes that make it possible to use sort of high-efficiency cold water laundry detergent. So a lot of laundry detergents have enzymes in them now that can break down stains on your clothes using enzyme that can kind of chew apart oils and proteins that are in stains. So you can use cold water instead. There's other sort of cosmetics and fragrances. There's also foods. There's some interesting work going on in plant-based meats. So a plant-based hamburger that has a protein in it that's able to make it look and taste and feel like meat, that red blood flavor. Yeah, it's called, it's Impossible Foods. They're great. So these aren't products made by Gingko. These are other products that have this kind of process in them. So Impossible Foods is making the veggie burger that bleeds using this technology, using yeast. It's delicious. I'm not sure. You can find them in restaurants all over the place. I'm not sure if they have them here, but yeah, check it out, Impossible Foods. Oh, do they have it? Okay. Check it out. There's also folks who are working with this kind of technology to make fashion materials. Fashion is one of the most polluting industries that exists, and there's people trying to make bioengineered leather. So this is leather made by making yeast that produce collagen, and then you can kind of create this liquid leather that can be shaped and formed into a lot of different shapes. This is from the Museum of Modern Art did a show last year on kind of fashion and technology, and this is one of the pieces in it. The company Modern Meadow is based in New York. Another company that's doing this with textiles is called Bolt Threads. They're up in California. They make spider silk in yeast. So they take genes from spiders and transfer them into those yeast. So now you can brew spider silk. These are garments made by Stella McCartney using that silk. And so the reason to use this kind of silk, spider silk is one of the strongest materials on Earth. It's biodegradable. You can produce really fibers and fabrics and textiles with really interesting and important properties for kind of performance garments that don't have the problem of things that are made with polyester, which is the microfibers that get washed away into the waterways. So there's some really interesting applications and really interesting kind of new technologies that are making their way into these everyday products via synthetic biology. Another project that we just announced at Gingko, we're partnering with a company called Kronos Group up in Canada to produce cultured cannabinoids. So taking the genes from cannabis that are used to make the active molecules in cannabis that have pharmaceutical properties, and then make them in yeast. And the reason to do that is because there's a number of different molecules inside of the cannabis plant. This is one of them. It's called THCV. It's present at really, really low amounts inside the plant, so it becomes impossible really to extract it in any economic way. So I think when you try to kind of calculate how much it might cost, it's looking like almost $1 million per kilogram of this kind of molecule if you're going to produce it for pharmaceutical purposes. But if you could take the genes that make that compound and put them in the yeast, now again you can brew these compounds and produce them in a much more economical and accessible way, make them possible to bring them to different products. Another product that I'm working on, is to actually use the same technology to bring back the scent of a plant that went extinct. So this is a photograph of a sort of preserved specimen of Hibiscadelphus wilderianus. It went extinct in about, I think 1912. It's a relative of the hibiscus plant that was growing on the lava fields in Hawaii. The botanist who discovered it and took those samples only ever found one sample, one specimen, and it was just kind of hanging onto the side of the cliff, and the whole habitat was shifting and turning over to cattle grazing. So especially in island areas, and as cities and humans encroached in different areas, we've lost a lot of plant biodiversity, and those preserved specimens, they do still exist in herbaria. So this is a photograph from the Harvard Herbarium where we found the samples. So each of those folders will have large pieces of paper that have the specimens kind of pressed in them. The Harvard Herbarium has about 5 million different samples, but and within there we were able to find about 15 different extinct plants that we were able to sample a tiny bit of material from, and then DNA, and then from that extracted DNA sequence, we were able to construct genes that we predicted would be able to produce a number of compounds that would produce a smell. And so from those genes, we put them in the yeast, and we were able to actually measure what those compounds that were being produced by the yeast that would have been made in the plant itself. And then this is also an art project. That's the science part, and then for the art piece is the interpretation of that list of molecules. Because we'll never know actually what the smell was. That's lost to time, because the way that the genes would all interact inside the plant, how it would all interact on the lava field, how it all would be, we have to now interpret that information that we did get from the science and put that together to tell that story of what that plant really would be like. And we worked with a smell researcher and artist named Sissel Tolas. And so here's a sampling of what it looks like in her process, and how she kind of mixes the different scent molecules to produce this perfume. I have 10 of her sketches for this project, which I'll be able to show at the salon tomorrow. So whoever's coming to that, stay tuned. So this connection between the combination of the art and science, the interpretation of the DNA and the translation of that into something that we can experience, that's something that I'm really interested in, and I think that intersection between art and science is especially important when we're talking about synthetic biology and bioengineering and nature and messing with it and all those different things. Sissel and I actually worked together now almost 10 years ago on a project called Synthetic Aesthetics, which was an academic project run by Stanford and Edinburgh University that brought together artists with synthetic biologists, asking the question, how will we design nature, and how can we design it well? What does that mean? Like, how can we kind of explore the thorny ethics and aesthetics of synthetic biology and what it means for the future of nature? And so Sissel and I were paired to start thinking about that back when I was a graduate student. So I was really thinking still about microbes and how they work together and how they make really interesting chemical compounds in the chemistry of microbes. And Sissel obviously is this odor artist, and a lot of her work is about challenging people to think about odors in different ways and experiencing the world via odors. And so for her, she's really interested in some challenging odors. So odors that you might push away and not want to engage with. She asks people to put away that emotion and engage with smells and think about it and decontextualize. So a lot of her work is actually producing body odors, which she would actually paint, nano-encapsulate and paint on walls and galleries. So people can experience those smells in a new context and think about how do we interact with each other via smell, and why do I think some things are good or bad, and what is a good or bad smell anyway. And so as we were working together and starting to kind of like, what are we gonna do? I don't know, body odor is gross, I don't know. So Googling body odors and the bacteria that produce them. So it turns out most of the odors that we would attribute to or think of as human smells are actually things that are produced by the bacteria that live on our skin. And so as we were searching and learning more about those bacteria that live with us and make these smells that are us, we were finding a lot of really interesting things happening. So anytime I would type in the species name of the skin bacteria, I'd get something about cheese. I'm like, well, that's odd. And so the more we looked into it, the more we realized, like, actually, there is a connection here, right? There's the cheese bacteria, the bacteria that makes cheese, that curdles the milk, makes all those cheese flavors. It's actually, all of them are very closely related to bacteria on the skin. It's why you might smell a Limburger cheese and think it smells like feet. It's because it's the same bacteria, or very similar bacteria that lives on your feet, between your toes, and in the cheese. This is an interview with a woman named Danielle Pergamont, who's a Spanish cheesemaker in the New York Times. We found this while we were working, and it was so interesting because she says, the reason my cheese is so delicious, says Miss Amieva, is my hands. She turned her meaty, calloused palms over for inspection. The natural bacteria in my skin makes the cheese more flavorful. So it's rare that you would see someone literally say this, but then when you actually look at how people make cheese, and how people are part of the cheese-making process or winemaking with your feet, there is a connection here that is bringing together bodies and food. And so you might be able to say, well, we are what we eat. I have a close friend of mine is actually a cheese microbiologist. He's a professor at Tufts, and his research shows that the fungi that are on the rinds of cheeses, you can actually kind of think of them as 20x concentrated feet. The maps of the cheese and the feet are very overlapping, and it's kind of more concentrated. So, on the other hand, too, we eat what we are, and so that connection between the two via microbes, I think that's really compelling. And it was like, there's something juicy there, right? We don't, like the armpits, we don't want it, right? We say that's bad, that's smelly, we don't want that. We push it away. But with cheese, we welcome it in. We want the stinkier cheese, the better in some cultures. And that, like, what makes something smell good or bad? What makes a bacteria good or bad? I think those are all like really interesting cultural and aesthetic questions that I think as we talk about bringing more bacteria into our lives via these products that synthetic biology is going to produce, are going to be really important for us to kind of deal with. Like, how if we can't think bacteria are gross if bacteria are going to be part of our medicines. We can't think bacteria are going to be gross if that's how we're going to be, we're going to have new cosmetics that are going to be bacterial that make our skin smell and feel nice. So we'd made our own cheese. We sampled some bacteria from our bodies. From between toes and belly buttons and ears and nose and mouth, and we made some of our own cheese. As artworks to challenge us and to challenge other people to think about some of these questions of what is good or bad, what is gross, and why is it gross to us? So I'd say it's food for thought. I did taste some. It tastes fine. It tastes like cheese. But it's not meant for consumption. It's meant for thinking. And that thinking, and with thinking that I think that art and design can kind of especially catalyze the questions that artists can ask via the work that they do with synthetic biology. I think it's really important, and it's important because I think these are going to be like cultural objects and cultural things that are part of the way we're going to interact with the world. The way that we think about nature, the way we interact with nature is going to be changing. It changes all the time. With climate change, we realize that nature isn't out there anymore. Nature is always embedded with the work with things that we're doing, the technology that we have. This is an example of another artwork that's by a designer who works on issues of synthetic biology. She was one of the curators of the Synthetic Aesthetics projects and a writer of the book. Daisy Ginsburg, this project, Designing for the Sixth Extinction, imagines engineered animals that are deliberately engineered and released into the forests and into nature in order to save nature. So a slug that will neutralize an acidic pH in the soil. Or if you look closely, there's all sorts of impossible creatures in the forests, like things that can kind of like filter the air and improve and change nature, but engineered and sort of already not nature. And it encourages us to ask these questions like, well, what is nature? What should it be? How will we interact with it? How do we already interact with it? How will people regulate this? How will people think about this? How will that change the future? So these are thorny and difficult questions I think for scientists to deal with when we're in the lab. So I've appreciated working with folks like Daisy and Sissel, and I think it's a really important way for us to think about how we talk about and think about the future of genetic engineering and GMOs. And so GMOs, I want to talk about GMOs. So like maybe you'll see I have this sticker here that says I heart GMO. Because I think GMOs are this flashpoint for talking about what nature is and how we think about nature and how we think about technology and where they do and where they should intersect. There's a lot of politics there. There's a lot of things that are really messy and complicated. And then there's this problem we have where, all these companies said like, oh, you don't need to know about GMOs. Don't worry, we're not going to label them. And so at Gingko and a lot of the work that I do is also like using that art and the politics to really open it up. How can we talk more about this? How can we actually talk about what we want the technology to be and what we want it to do and why things are engineered the way they are. So we wrote a New York Times article a couple years ago pushing for GMO labeling. We're proud of the technology. We're proud of the work that we do. We think that this is being used in really positive ways and can be, and it's something that people should know about and be able to decide for themselves. So we pushed for labeling. We also worked on designing new labels, imagining what it might look like if you did have a label on your on the products that have those new fragrances or new enzymes or new cultured products, what that might look like. So you can kind of talk to people about how they would interpret those labels, what they want from their products, what the kinds of ingredients they want to be seeing. And then we're hoping that we can open up a conversation about why would you like GMOs? What could it be? This is not to say that I love all GMOs and all the way that it's always been used, but I want people to think about the technology as something that could be lovable and what that might be. So that's something that I think that the art and the design, I think, lets us ask those questions, lets us open up those spaces for debate in a way that kind of gets us outside of this like pro and con only, and that's kind of intractable debate that we have right now when we're thinking about the way technology is going to be part of our lives. So I can go and in my work, I've tried to encourage more ways that we can work with artists and with designers and ask those kinds of questions. So we've created a residency, a creative residency, bringing creative people, designers and artists into our company that's full of engineers, so that we can be having these conversations on the ground as we're working, and that we can be creating new kinds of projects and stories that can ask new questions about what this technology is and how it might look in the future. So our first resident was Natsai Chiesa. She's a designer. She trained she has a master's in material futures, and she's a textile designer. She thinks a lot about materials and what she calls the post-carbon material future. How are we going to incorporate biological things and decarbonize our materials and the things that we interact with? She runs a design consultancy called Faber Futures, where she asks these kinds of questions and collaborates with companies to prototype new kinds of products and materials. One of the projects that she had been working on for a few years and was kind of interested in exploring in the context of Gingko, in the context of synthetic biology that can happen in our facilities, was this project of looking at dyes produced by bacteria. So these are all the soil bacteria that she was able to collect from her garden and identify different pigment-producing species that make a huge range of different colors. So bacteria naturally produce these kinds of pigments in the soil. They can act as sort of antibiotic molecules and kind of communication molecules in the ecosystems of the soil. And so depending on the acidity of the soil and the acidity of the media, there's some molecules that'll turn really bright blue and some that'll turn pink, and you have this really interesting range of colors. And so as a textile designer, she found that really interesting. Like, could she use these molecules? Could she use these bacteria themselves in the process of dyeing those fabrics? So she was particularly interested in how she might let go of some of the control that she had as the designer to sort of control how the pigments were placed on the surface of the bacteria, of the silks, and actually give up some of that control to the bacteria, to nature, as a co-author of her designs. So she developed a number of techniques to bring those bacteria into this process of shibori dyeing, where she was able to kind of create a number of different kinds of patterns and colors on sort of small scale silks. The bacteria are producing pigments that are color fast. You can wash the bacteria off and they leave the bacteria behind and they last through washing. They don't need the same kind of mordants and chemical additives that you need to have traditional dyeing, and you need much, much less water. So I think she calculated for a kind of t-shirt size piece of silk, she would use about 200 times less water than a typical method that is used today in industry. And so she was able to, like with the petri dishes that she had in the lab that she was working in within London, she could kind of make almost like 2-meter size pieces of fabric. That scale was about as high as she could go, and it was all very bespoke and handmade. And she was really interested in this intersection between kind of the industrial processes that are starting to happen in biotechnology and the craft practices that she could do herself as a designer, and ask questions of how that might scale and what that might look like as if this were to kind of approach an industrial process. And so when she was in the lab at Gingko, she explored a lot of these questions about process, about kind of expanding and scaling up, looking at the robotics and what we were working with, looking at the engineering of the strains, how you might be able to engineer to produce more or different kinds of pigments. And then she was able to, with a lot of work went into this. This is the world's first bacterial dyed garment, the first caftan dyed with bacteria. And with this material, with this fabric, with this garment, ask some of those questions of like, well, what is the future going to look like? Are we going to be able to have as much clothes as we have if we're dyeing all of it with bacteria? Are we going to be wanting to throw away clothes the way that we do now? Are we going to have different kinds of cycles and different kinds of economics? Are we going to have a different kind of scale of these global resources as these things kind of come online? So tricky questions, interesting questions that are kind of catalyzed by these also really beautiful objects that I think show you something that awesome that bacteria can do. We're about to start our next residency with Yasmine. She's a designer and is actually an interaction designer. So she worked on the operating system for Microsoft HoloLens, which is a sort of AR, VR goggles. So she designed the systems where if you're wearing these goggles, like how you would interact with the computer and interact with the world via these glasses. So she's really interested in perception, interaction, and the senses. How can we use technology and these kinds of different tools to expand our experience of the world? More recently, her work has focused more on smell and taste and the biological senses. And so she's actually really interested in something like this, these biosensors. How can we create new kinds of sensing apparatuses that are able to sense different things about the world? How can we use these kinds of systems and design them to be able to learn more about the world around us, and how does that information translate to us? I think that's also really important because when you think about biotechnology, right, this is impossible to understand, right? Like no one looks at this and says like, yes, it's a I know what this is sensor, or what that even means, right? Like the molecular scale is so intangible, it's so inaccessible, even to people who spend 20 years thinking about the scale. So thinking about how we might make sense of this and experience it as humans every day, how that might change our experience of the world and our interactions with it, how that might change the way, our own interactions with our own body and the world around us. I think there's something here too. So stay tuned. She's starting in October, and we'll be posting. There's a creative residency Instagram, which I should have put. But anyway, you can follow along. I want to close with this. This is an idea. This is a statement from Drew Endy, who's one of the founders of synthetic biology. He was a professor at MIT in the early days of the field. He's trained as an engineer, and so this question of like how engineers would work with biology is really what catalyzed this new field, this new way of working with biotechnology. He's now at Stanford, and he was one of the leads on the synthetic aesthetics project. And he's been, I think, always very thoughtful about how biology is going to be part of technology, and how he as an engineer would interact with biology. And I really like this line and this thing that he says. He says, I wish for humanity to transition from living on Earth to living with the Earth. And I think, when we think about nature, we would often think about nature as outside of us. We're on Earth, nature is far away. Nature is not something that's messed with or meddled with. But how can we kind of reclaim the fact that we are part of nature, we are nature, and that we need to live better with nature? How can we, what's that gonna look like? What's that gonna feel like? What do we need to do to get there? Those are all really big questions that I think folks like Drew, all the artists and scientists who are working together in this new and changing and growing field of synthetic biology are trying to figure out as it comes out into the real world. So there's lots to think about, lots to do, and I hope that you guys keep asking questions too. So with that, I would love to take your questions and hear from you.